Hello everyone. Welcome to the October 23rd Biocurator Working Group call. Today, we're really excited to have the Low Penetrance Risk Allele Working Group here to give us some updates. Um, on all of their hard work. And then this is the last meeting we're going to have for quite some time because of holidays, at least in the US. So both November meetings are going to be canceled. And so the next meeting, which will actually be the last meeting of the year, I can't believe with the time how it passes, is Wednesday, December 10th. This one will be the Wednesday, 4 p.m. Eastern time one, which will be a celebration of curators where Courtney Thaxton, our steering committee chair, will just talk to us about all the progress that you've all made as curators in the last year in ClinGen and give a little update on that to celebrate you. So, with that, I will turn it over to you, Ryan, and you can share your screen.
Okay. All right. Well, hi everyone. First off, I'd like to thank all the biocurators. You guys do all the real work in ClinGen. I remember doing some of that work myself. Let's see. You guys are seeing my full screen PowerPoint now?
Yep,
Okay,
looks
great.
good.
So I am Ryan Schmidt, and I'm here with Matt Lebo and Marcie Steeves. So the three of us together, we're the leadership of the ClinGen Low Penetrance Risk Allele Working Group, and I'm going to give kind of a overview of what we've done in the past several years and what our group is working on now. And I'll just say in advance, a lot of our work centers around harmonization with how we deal with these variants that have lower penetrance. So I think this can be interactive today. If you want to kind of pause and stop me for a question, that's totally fine. I might not be able to see you, so just go ahead and speak up. And hopefully we have time at the end to have a little discussion and answer questions. So with that, let's get started. You're probably sick of seeing this figure, but I think this helps illustrate what our group is interested in. We're interested in these variants that have kind of intermediate to low penetrance, so lower than we usually think about with our higher penetrance Mendelian variants, but it's still enough penetrance to be clinically significant, where the variant alone would appear by itself on a clinical report and maybe be tested for by a clinical lab. And these variants, as you might know, they frequently have population frequencies that are higher than those that we usually deal with for our Mendelian disease variants. All right, so why are these challenging? And many of you have probably encountered these variants in your work as curators, in your disease area expertise, and I'd love to hear some of the individual stories at the end. But let's go through why they're challenging in general. So again, frequently they're more common in the population than expected for the disease. So that kind of triggers benign evidence criteria, and that can lead to conflicting evidence. These variants may not be able to use traditional evidence types for classification. We'll get into that a little later. So you kind of can't take advantage of evidence that might lead you to say this is disease causing. It can be difficult to define when increased risk is clinically significant. We've observed a large rate of discrepancy in ClinVar and other places between laboratories and curators. And then I'll say previously, there was no universally agreed upon terminology for even describing these variants. And I'm going to tell you a little bit about some of our work, and hopefully that can lead to a little bit more consistency with how we talk about these variants. So this slide here is kind of like our conception of the universe, and let's just walk through it from top to bottom. What we're doing is introducing two new variant classes. So most of us probably in our day-to-day lives might operate over here in this high penetrance space where the absolute risk of disease, absolute penetrance is going to be closer to 100%. And we know that we can classify those variants quite well through our standard ACMG framework and get to these different criteria, or sorry, these classifications down here. What our group has done, and I'll go through more in subsequent slides, is carve out these other two areas of the universe. So now the spectrum of penetrance of variants can be separated into what we call risk alleles and low penetrance variants. So one thing I want you to take away from this is that these are very specific terms with specific meanings that we arrived at through consensus building within our group, community surveys. So there was a process, and when we say these words, they are referring specifically to these classes of variants. So let's take the risk alleles here on the left first. So they have quite low penetrance. The genetic evidence type that predominates with them is association studies. And one thing I'll show you in subsequent slides is a risk allele classification framework that our group developed. And that framework leads to these classifications that you see here: established risk allele, likely risk allele, and uncertain risk allele. And the nice thing is that those kind of map with the level of confidence we have in the classification to what we're used to with pathogenic, likely pathogenic, and uncertain significance. And by the way, there is a concept of protective alleles, which we don't encounter as often, but it's the same thing as a risk allele, but it's protective against disease. So it's kind of the magnitude is in the opposite direction, but this framework can be used to classify those variants. So that's the risk alleles. And then low penetrance variants is something that's in between. It may share more features with the high penetrance variants, and that's something we're working on now. And we would propose that you can generally classify these in the Mendelian framework, but with some modifications potentially. All right, so this is a publication from 24 out of our group. And what this publication does is, sorry, I jumped ahead. It does three things. So one, it lays out the terminology that I just discussed with the risk alleles, the low penetrance variants, and those different classifications. It provides a risk allele classification framework. And then lastly, it discusses some reporting scenarios that you might encounter with risk alleles. And we sought to see if consensus could be determined internally within our group about how to handle those scenarios. So just some thoughts on how to handle those scenarios. All right. Um, so here are these terms, and what you'll see is that the definitions are relatively qualitative. So a risk allele, it is going to potentially have an appreciable population frequency. You're not likely to see it segregate with disease in families with a clear inheritance pattern. And the additional factors are going to be bigger determinants of whether or not you manifest the phenotype. So there's going to be additional environmental and genetic factors that come into play. Again, the risk allele is supposed to be something that alone is clinically significant and might appear on a report. Let's contrast that with the definition of the low penetrance variant. This may show some evidence of segregation with disease in families, but the penetrance is reduced such that the variant alone is no longer deterministic as to whether disease is going to occur. As I said, our group is there to do harmonization and consensus building. And we can only do the best we can with the level of consensus we currently have in the field. So what we're able to do is get to these qualitative definitions and also kind of survey the community about where they thought the line was if you were to put a number on it in terms of the amount of penetrance. That's tough to do because in our world today, we don't always have highly accurate estimates about variant level and gene level penetrance. But let's imagine that you did, and we asked people to kind of tell us where these lines are, kind of represented by these gray zones here. And what they told us back in these surveys is the line below which you have risk alleles is something around 10 to 20% penetrance. And then for the low penetrance variant, it's potentially below 50%. And we've heard feedback after this being published that, well, for some disease areas, they feel like maybe this number is too high because a lot of what we're learning now is that our variants aren't as penetrant as we thought, or some disease areas like cancer predisposition tend to have lower penetrance even for their higher penetrance variants. So it's all very challenging to kind of come up with something that can accommodate everyone, but this is what we have got to at this time. So just for everyone's information, these terminologies and classifications are available in ClinVar. So that can be very helpful when uh, you can't get to the classification you need using the high penetrance classifications, because you'll see, as you probably have seen, variants in ClinVar where the same variant is marked benign, likely benign, VUS, likely pathogenic, and pathogenic. So sometimes these other classifications can be much more helpful and lead to consensus. I think I have some numbers here, but these are outdated with how many variants are in there at this time with these classifications. Um, so with that, I will launch into a little bit more detail about the risk alleles, but I think maybe why don't I just pause for 1 second in case anyone has any questions they want to ask right now. All right, I will continue on, but feel free to speak up and happy to be interactive.
Ryan, one question.
Oh yeah,
Just is the terminology finalized or still on discussion? So
final. Yeah, that's that publication. But yeah, go ahead, Matt. Yeah.
yeah, so we have sort of finalized it, and it's part of, as Ryan was mentioning, part of ClinVar now. Um, the sort of the exact buckets, as Ryan was saying, of when you go from one to the other hasn't been finalized, but the actual terminology has. And we did a lot of some community engagement and internal discussions to sort of get to that point. So.
Mhm. Thank you. Well said. Okay. So now, again, there's these three different portions of the universe, and I want to talk specifically about the risk alleles and the framework that comes from that. So this is in that publication, by the way, if you want to kind of look into more depth in that. So one of the first things in the publication are some recommendations with regard to the curation of evidence surrounding risk alleles. So that's highly relevant to this group. I can't really go into all the fine details in the publication, but just let's hit the highlights of what's important to think about when you're dealing with a risk allele. And keep in mind that the association studies are going to be the predominant form of evidence here. So it's critical to determine uh, whether the disease phenotypes are consistent across studies and with your disease of interest. So phenotyping in the studies is important and how that was done. Um, Matching cases and controls is critical because that can lead to kind of a false positive association because of the presence of bias. So studies should match cases and controls based on ancestry, environmental, demographic, and any other confounding variables. If those things are not matched well, the quality of the evidence coming out of the study is essentially lower. Um, the one note here is how the variants were detected. So if you have different technical methodologies that detected variants between cases and controls, that can lead to some type of bias. It's important to recognize the genetic model that's used. So sometimes in case control studies, they will use a dominant model, they might use a recessive model, or just kind of like an allelic model. So it should be clear and consistent which is being used. And then statistical significance. Of course, many studies nowadays, they're looking for the association of many variants at the same time with disease. So it's critical that appropriate multiple hypothesis testing is performed because otherwise that's statistically significant result is in fact not significant. Um, and then separate from the significance is the strength of effect. That's usually calculated in the odds ratio in these studies. Um, but you really shouldn't accept any assertion of an effect unless the result is statistically significant. And then the last thing is replication. Essentially, for it to be a clinical grade association, we really want the result to be able to be replicated in independent populations. All right. So I don't want you to read all the words on this slide, but just keep in mind that within this framework that's proposed in this publication, that there's a set of criteria, and then those criteria lead to classification. So similar to what you're used to in the high penetrance system. But note that this is really simplified and driven by association study evidence, and we'll go into that in a second. But the output of this is essentially a three-tiered classification towards a risk or a protective effect. So established, likely, or uncertain. There is a recognition that you can have a refuted association in some cases. Um, so again, this framework can be used for both risk alleles and protective alleles. And the evidence types are association studies and functional studies. So functional studies are supporting evidence. You're not required to have a functional study to tell you the mechanism of how the risk allele is working. It's really driven by the association study. And in fact, the risk allele doesn't actually have to be the functional variant that is mechanistically causing disease. It could be genetically linked to some other potentially unknown variant. All right. So for the association study evidence, the for the criteria, you can achieve uh, limited or conflicting evidence, strong evidence, or very strong evidence. And each of these criteria looks at three elements, which are shown in the bullets here. So the number of individuals in the study, whether the results are replicated, and what is the risk of bias based on the methodology. So let's consider the very strong case. We're proposing a floor of that this is something where there's many individuals studied, so over 500 cases, over 500 controls. There's extensive replication with little between study inconsistency, and then bias could not reasonably affect the presence of the association. Let's contrast that to limited or conflicting evidence. So by the way, to get very strong, you need all three of those things to be true. So for it to be limited or conflicting, one of these things is going to be true about the variant. So there's a limited number of individuals studied. There's lack of replication or large between study inconsistency. Essentially, could be conflicting evidence, or there's methodologic issues and there's potential for bias. Strong evidence would be somewhere in between. For functional studies, again, it's very different from high penetrance variants where functional studies can give you strong evidence. This is really supporting evidence that doesn't move the needle in terms of classification, but it kind of gives you additional confidence because someone suggested a biologically plausible mechanism for the effect. And something we'll get to later is that for these variants with lower penetrance, um, the traditional functional studies may not be geared to detect the effect. So they may require appropriate controls that variants with lower penetrance to detect subtle effects in some cases. Um, for refuting evidence, so this is a scenario where an assertion has been made at one time, but now there is essentially overwhelming evidence that that assertion is not true. Um, so what's going to happen is that there was a smaller study or studies that is refuted by a very large and well-designed study. But it does get tricky because what we talked about earlier, the definition of disease, how you phenotype the individuals, the fact that different ancestries can have different ancestry-specific effects. So all those things have to be taken into account. But there are some scenarios where an assertion has been made and then later refuted, and it's not just disputed that there's kind of like a equal evidence against it. It's that there was a small study saying this is true, and then a massive biobank study says it's not true, for example. Um, Ultimately, you lead to one of these classifications that I'm putting up here. It's really driven by the association study evidence. And keep in mind that if there's no statistically significant association study, there's no genetic evidence for the risk allele, it would remain unclassified. If that were not true, you would have a classification for every possible variant in the genome. That's not how this works. It's that once an assertion has been made that's statistically significant, then it can go into the system and be classified. Um, were there any questions?
No, nope, not right now.
Okay. Um, and then the last thing from this publication were these consensus opinions. So I don't want to overstate what was done, but what the group did was within our working group, which has people from different disease-specific and variant-specific working groups across ClinVar, we considered different scenarios and talked about how you might handle the reporting of that and tried to see if consensus could be achieved. So you can see these scenarios in bold where it's kind of like a diagnostic, think about like an NGS panel for the same indication, meaning that here's an APC variant. So they were getting a panel for cancer predisposition. And now this variant was found and it is a risk allele. Would you report that or not? And our group came up with the consensus that yes, they would report that, but you wouldn't in your report kind of have the same top line as positive as you would if you had a Mendelian variant. So in some regard, the risk allele should be handled separately or put in a separate section to show that to appropriately communicate the amount of risk that's actually associated with the variant. In contrast, here's an example where they had a gene panel for hearing loss, but then they found a risk allele that's associated with melanoma. So the group, I think there's no consensus to report that variant. So we went through all these different scenarios, including prenatal, secondary finding, healthy genomic screening, and carrier screening, and we just talked through some examples, and we tried to see if we could come up with a consensus. So we put that out in this paper in case that's helpful to the field. So with that, now we're going to change gears a little bit. We're going to talk about low penetrance variants. I'll stop for questions again if there are any.
There was one that came through in the chat, Ryan, which
Okay,
you like
let's
kind
let's
of
do that.
answered, but how do you
Okay.
know when it's appropriate to use the risk allele framework versus the typical Mendelian framework?
Yeah, great question. So yeah, that's the one we are trying to answer and give as clear an answer as possible. But again, based on the what we know about variants now in terms of the how well we can estimate their penetrance, we can only kind of give a qualitative answer. So it is very tricky, and a lot of it has to do with whether the evidence types available uh, tend to fit better in the or other of these systems. What we believe the penetrance of the variant or the penetrance of variants in that gene to be. We talked about those rough cutoffs. But yes, that's now a consideration, right? Because you have to decide which one to kind of go with. So perhaps you just use your best judgment and you see which one fits the most based on the uh, evidence you have around the penetrance and the genetic evidence type, for example. All right, so by the way, we changed gears. Now we're talking about this group of variants in the middle. And the risk allele stuff I just told you about, that's previously been published. This work, what I'm telling you is ongoing work or work that's been put out in a white paper on our ClinGen website describing the challenges of classifying these variants, but we haven't put out any formal recommendations on how to deal with those challenges. So that's something that's a work in progress. And just to give you kind of a hint of our group's thinking, we believe these variants are more similar to the high penetrance variants, and they are likely to be able to be classified in that framework with some modifications. And by the way, we have these classification terms that we recommend people to use. All right, and here's that white paper. This is just a screenshot from our website. So if you say go to the Low Penetrance Risk Allele Working Group website, then you'll see a link for this white paper, which is a PDF that we put out there in March 2025. All right, so so we're talking about low penetrance variants, and what a lot of the work our group's been doing has been looking through the different categories of evidence for the high penetrance variants in the Mendelian framework and trying to see which ones are relevant potentially to low penetrance variants and which ones might be challenging for low penetrance variants. So where does that system break down essentially for low penetrance variants? So the first set of evidence types we'll talk about here is genetic evidence. And the new term for that kind of in the upcoming framework is human observational data, but kind of in the same space here. So let's talk about one that's relatively easy to wrap your head around, population frequency. So we have in the upper right corner, we have some of the current evidence codes, and then we have the upcoming criteria, the observation for population, the abbreviation there. Um, and the issue, as we mentioned before, is that because the penetrance is lower, these variants can have a population frequency that's above the allowable threshold um, for a certain disease for a certain gene, and that can trigger benign evidence criteria. Um, and essentially, the solution is to have a scenario where you can discount that benign evidence criteria due to the high population frequency when there's sufficiently strong genetic evidence of pathogenicity. Um, but that's under discussion how to figure that out. What constitutes sufficiently strong genetic evidence. Here is a figure from the upcoming framework. And in that framework, you get penalized. You have these negative points when the population frequency is above certain thresholds, which are multiples of the disease allowable frequency, um, disease allele frequency threshold. And you get these negative points. And we believe that the low penetrance variants have the potential to fall into this space and potentially inappropriately receive -1 or -3. And thus, there needs to be a mechanism to kind of disregard that. Proband counting. So for very rare variants, proband counting can be an important evidence, a piece of genetic evidence to establish pathogenicity, right? You have these rare variants, you have publications where they just list out the variants that they found from certain testing. And, uh, there are some specifications that have come out that say, well, you can't use proband counting data if you can't apply the PM2 evidence criterion. So essentially, if the variant is too rare, you can't, sorry, too common, if the variant's too common, you can't use the proband counting. And that's appropriate, but it's important to note that low penetrance variants, um, they're ineligible frequently to receive that evidence of pathogenicity. So in some ways, if you think about it, they're kind of discriminated against, or there's one less pathway for them to get to pathogenic, or it's harder for them to get to pathogenic. So thus, does that mean there needs to be kind of like, and you'll see the same theme. It can be harder through other evidence, different types of evidence to get to pathogenic for low penetrance variants. Do we need to have a lower bar for these low penetrance variants so that the ones that really are disease causing can get to pathogenic? Um, for segregation evidence, you can certainly use segregation evidence in families for low penetrance variants, but the thing to know is that there's going to be individuals that have the variant but are not affected with disease. So those would just kind of be ignored and not counted. And what that means is you ultimately need a larger family to to achieve the same strength of evidence as you would in the high penetrance system. We've noted that there are some recent kind of more advanced methods that have been developed that can help in analyzing pedigrees for low penetrance variants. But again, this is just another case where the the variant it's more difficult to get to pathogenic for these low penetrance variants because not all of the individuals will be counted as affected. For case control studies, it's possible that the controls are contaminated by these non-manifesting variant carriers, right? Because they don't have the disease, but it's a low penetrance variant, but they're going to be stuck in the control group. So ultimately, it leads to a lower effect size. It's not as significant. So again, the low penetrance variant will receive reduced strength of evidence for pathogenicity, all else being equal. Or, it will require a stronger effect to receive the same strength of pathogenicity. All right, so those are our thoughts on genetic evidence for low penetrance variants. Should we take a break for questions or keep rolling?
I think you can make it through this one and then we can break for questions. You've been doing a pretty good
Okay.
job breaking.
Okay, I promise the pain will end soon. So, but it's okay. We're talking to ClinGen, so you guys are used to this. All right. So now experimental evidence, which is now the politically correct term is predictive and functional data. So computational prediction. So, computational prediction for splice region variants, for missense variants is important. Um, it has provided supporting evidence in our current classification system, but is eligible for even stronger strength of evidence in the upcoming proposed framework. And it's unclear if low penetrance variants as a group obtain lower scores from computational prediction tools relative to high penetrance variants. That's something we're trying to determine. But it's important to note that when people developed computational prediction tools, they frequently held out low penetrance variants. That's something you don't want to kind of mess up your training with if your computational prediction tool is geared to detect high penetrance variants. So they may have been excluded from the set of disease-causing variants, and they, in some cases, may have contaminated the controls. Um, it's also important to note that some computational prediction tools incorporate population frequency, which could, again, work against a low penetrance variant. And then finally, it's possible that the low penetrance variant has kind of a partial loss of function. So it may only partially disrupt splicing, and that may be associated with intermediate score outputs. So, what is the solution? To be determined. We're further investigating to what degree computational prediction scores differ for low penetrance variants. But in an ideal world, we know that it'd be better to kind of calibrate the computational prediction tool with a set of low penetrance variants. And you'll see a parallel uh, thought process coming up for functional studies as well. Um, Enrichment of pathogenic variants in a functional domain. So, there's recognition in our current frameworks that if you have a region of a gene of a protein that really doesn't have benign variation, if you see something novel and rare in that region, we can say that at least provide evidence that that is a disease-causing variant. So it's interesting. There are some examples of genes where variants in a specific domain are associated with low penetrance. So the question is, can we conclude that a novel variant in that domain is a low penetrance variant? So that's one kind of pathway where we're discussing. For functional studies, it's kind of similar to the computational prediction tool that the variance might have been excluded when kind of developing the cutoffs for the functional studies. Um, they might be inappropriately included in either the benign or the high penetrance pathogenic reference sets used. It's also possible that the functional impact might be reduced. So again, in an ideal world, you calibrate your functional study for low penetrance variants in that gene, and that would help us to develop thresholds to appropriately classify a variant as low pathogenic, low penetrance. So again, in an ideal world, we could have a novel variant, run it through the uh, functional study and then have a strong indication of whether or not that variant's going to cause disease and whether it's going to be low penetrance. But that might not always be possible. Um, Last couple of concepts that our group has come across in our discussions. One is the low penetrance gene. So there's kind of two scenarios. The most common scenario is that you have a gene that normally operates with high penetrance. It's kind of a Mendelian gene with high penetrance variants, and then you have a rare number of variants that are different than those high penetrance variants. They cause disease with low penetrance. So that's one thing. It's also possible that all the pathogenic variants in that gene cause disease with low penetrance. So something like a CHEK2 or something like that. It's lower relative to other genes in the same disease space. Um, so so that changes things a little bit because the this maximum allowable population frequency threshold, it's based for those genes, it's already based on variants with low penetrance. So the problem with the population frequency is potentially less of an issue. Um, but yeah, this concept is something we've been kicking around, whether or not there's a pathway to um, classify your gene as a low penetrance gene, and then, novel variants in that gene would kind of be assumed to be low penetrance. But again, to do that, you have to come up with how do you call it a low penetrance gene? You have to come up with kind of criteria for that. So, but anyway, just a concept we've been kicking around. And then uh, in our discussions, we have also stumbled across a variety of other terms and concepts that These were not formally defining like we were with the risk allele and the low penetrance variant. So I want you to take away that the risk allele, low penetrance variants, those are very specific terms that we did a lot of work in consensus building to define. But then there are all these other terms you might hear in the space that are, tangentially related or not related. And in this white paper, we just kind of list many of these terms and give a little explanation of how we refer to them. But again, these are not formal definitions. They're just other concepts that you might come across, and we just wanted to kind of compare and contrast them and give a definition for how we talked about them as a group. So with that, I want to acknowledge all the hard work of everyone in the group, both for the risk allele work and the low penetrance work. Want to recognize all the contributions from outside the group at the ClinGen level, steering committee, all of you all in the community. It's really amazing the amount of things that ClinGen is able to accomplish with everyone's input. So. With that, I will stop and we can have a little bit of discussion. Thank you.
Thanks so much, Ryan. That was great. People feel free to stick questions in the chat or unmute and ask questions. In the meantime, Matt kind of answered this in the chat, but I know we already talked about the confusion of, reduced penetrance variants in a high penetrance gene, and then also variants in reduced penetrance genes. Someone had also asked about susceptibility genes, and if you guys are thinking about susceptibility genes, and Matt had said maybe those would fall along the reduced penetrance Mendelian spectrum too, but love to get your thoughts or just talk about it out loud.
Matt, do you want to comment a little bit on the susceptibility? Yeah,
Yeah, so I was thinking things like
yeah,
G6PD and malignant hyperthermia. And so in
yeah.
which case it's like the penetrance is somewhat high if you have exposure, right? And so I think
Hmm.
those were likely to follow. So and we might use some of the concepts or recommend some of the concepts we use from our low penetrant work, and we're still, like I said, trying to go through specific variants and test these out and see how they'll fall. And we have talked to, well, I've personally talked to like both chairs of those two different.
Yeah, that's good. I was going to ask because they do both have VSEPs at this
Yeah.
point.
Yeah.
So, and I
We,
think there are
we've
probably others.
largely been hands off with that because of the strong essential
Hmm.
Yeah.
environmental or exposure component.
Yeah, and I
Yeah.
think in both those instances, they're almost more curious about less of which framework to use, but how to actually like what term to stick on the variants. And
Hmm.
so I think we'll have some discussions with them for sure about some of those things. But it is, I assume if that's not what you meant by susceptibility and you want to talk about something else, we can chat about that. But in terms of like that type of like known environmental exposure type ones, that's sort of where we're falling. There are slightly there's a KCNE one, I think it is, like drug-induced arrhythmia one that's a little confusing that we're like is definitely part of stuff we're talking about. So there are others that are like
Hmm.
sort of in this realm that we're trying to figure out how to deal with those ones. And they're a little they weren't on that list of other terms Ryan showed because they're sort of related to what we're doing, but maybe they're I don't know. So we're still trying to figure those ones out.
Yeah. Thanks. That's helpful.
Yeah.
Do others have questions?
Um, Along those lines, I guess I'm just curious about, so we talked about susceptibility, which has those environmental inputs, but also what about predisposition, right? In the cancer space. So like what does a low penetrance predisposition look like?
Well, I mean, yeah, the even today within the cancer space, there's recognized to be kind of low, moderate, and high penetrance genes. So different genes might have different penetrance associated with them. I think the rub with the cancer space is that their high penetrance ones might be kind of lower than other disease areas. Although even across all disease areas, when we over time we get more knowledge, things are lower penetrance than we thought at the high penetrance level. Um, so yeah, cancer is tricky because there's an environmental exposure component. There's the fact that the pathogenic variant can lead to different cancer types. So if you think of like BRCA, you have a certain penetrance for ovarian cancer. You have a certain penetrance for breast for your whole lifetime. Um, so that gets tricky. But yeah, they're tougher. I think for cancer predisposition, they have found it tougher to swallow this, if you will, because they have a lower penetrance to begin with, and then they feel uncomfortable potentially going in the room and telling someone that you have a pathogenic low penetrance variant or something like that. But I really don't think they should be uncomfortable with that. I think our whole goal as a group and as a field is to, as accurately as possible, convey the penetrance that comes with the variant. You know what I mean? Ideally, we can have these clinical reports and like tell someone, with a high level of precision, like this is the exact risk and put a number on it if we could. So I don't think it's a bad thing even if the variant is like it's clinically significant, right? I mean, you could have a low penetrance variant, but you should still do screening or you should still do some intervention. So I don't think uh, people should be scared off of the terminology. But some people in the cancer predisposition space, they those are the issues that come up. They're saying that, well, even our high penetrance stuff is starting to encroach on the low penetrance area, and we don't like to call anything low penetrance because we are worried that people will walk away with the wrong impression. But I'd love to hear people's comments that work in the space. And again, please don't shoot us because we're doing the best we can, trying to get consensus across all these different disease areas.
So we have two more questions in the chat. Maybe I'll go to the cancer one first and then we'll hop back to the other one. So on a related note for cancer predisposition, what if you have a low penetrance variant and a higher penetrance cancer gene so that the person's risk is potentially much higher than a high penetrance variant in a different gene?
Yeah. Yeah. So the thing for that is that's why when we introduce this, it's based on absolute penetrance. It's not based on relative penetrance to what your high
Mm,
variance. So basically, let's say we knew the lifetime risk, the absolute penetrance. We would just kind of come here and be like, it's 20, whatever, 25, we're going to go right down this path. Whereas you don't have to be like, well, what's the high penetrance number for that gene? And compared to that, it's just like that's why we're talking in terms of absolute penetrance. And as I mentioned earlier, we did a lot of brainstorming about the future when you're going to have genomes on everyone and have more accurate numbers about things. That's the kind of world we want to operate in, where you could have a report and come out with their penetrance, estimate based on the data.
And yeah, and
It's
Ryan,
not a relative thing. Yeah.
like this sort of alludes to what Ryan mentioned too, this like low penetrant gene concept, and that is something that we're obviously very much aware of and trying to work through, like how the terminology around variants in those genes should be represented. So we will have guidance on that too.
great, thanks. That's helpful. So let's go back to frequency because I know we talked about that. Frequency BA1, BS1, PM2s really just are not working for risk alleles. And I think there's always that big concern of what if you accidentally filter out a risk allele just based on frequency. So this question is, are there any estimates for the frequency of low penetrance or risk alleles in general? How cautious should we be in classifying variants by the Mendelian framework versus the others?
Yeah. Well, for risk alleles, the population frequency doesn't matter. So that's, as long as there's a association study showing the effect, that's good. Okay, for the
But you typically need enough population frequency to have power to show the effect. So it's a little
right.
circular,
Yes.
but
Until
Yeah.
we get to
Yeah.
the
Yeah.
world where we have like a billion
yeah.
genomes sequenced.
Yeah. Yeah. Yeah.
Okay, so then population frequency for low penetrance variants in the upcoming system, you can be penalized for if your population frequency is quote unquote too high. So here's the thing. The low penetrance variants we know about in today's world tend to have high population frequency. There's a bias
Hmm.
towards that, right? Like we know about it because there's a lot of people out there with the variant, and there's enough evidence. Maybe there's a case control study or something, but there's a publication around that. Whereas it's also possible that many of these rare variants that we see, they could actually be low penetrance. It's just that we don't know that because they're so rare. But in the future world, maybe where those things that are rare today there's enough evidence to say that they're low penetrance. So I don't think I directly answered the question, but those are the issues that we're thinking about. So a risk allele population frequency doesn't matter. You just need the association study. For low penetrance variants, they could have a very high population frequency. They could be very rare. It's just that it causes problems when the population frequency is high.
So I guess related to, potentially missing risk allele variants or if things are high frequency. And I know we've talked about this a little bit in like our policy discussions for like the VSEP management working group. Are you guys considering at all potentially, putting together classifications and submitting them to ClinVar to bring some clarity for some of these? I know that you're actively working with some VSEPs potentially in the genes that might be higher penetrance that already have specifications. But
Yeah.
get your
So
thoughts on
that's
that because I know they're
that's
highly conflicting in
right.
ClinVar, which just really
Yeah.
makes it a mess.
So what our efforts are trying to promote consensus and harmonization, but we're also not trying to step on anyone's toes or usurp the role of the gene or disease specific groups. They know more than we do about these things. So who are we to classify their variant in a different way than they would? So
Hmm.
we're not a VSEP. Um, but we would consult with any group to try to help to share what consensus we've been able to build in our group and to provide essentially options and recommendations of how to apply this stuff for your particular gene of interest. So our goal is harmonization. Our goal is for as many people to kind of use these terms and these pathways as possible so that as a field we get more on the same page when we don't have you don't go in ClinVar and it's like same variants like benign, VUS, pathogenic, you
Yeah.
know.
Yeah,
Yeah.
and the one sort of addition to that. So when we did publish our risk framework, we were asked to show it for some example variants, and so I think that will likely be the case for the low penetrance framework as well.
Mhm.
And so in that regard, we will have classifications from our group and we will probably choose those ones smartly of like which ones are, and maybe they're ones that are commonly conflicting. Um, and we have discussed potentially about submitting those to ClinVar, even though we're not a VSEP. So it wouldn't get like
Hmm. The three
star,
stars.
the
Yeah.
three-star classification, but we would submit it because we did the work and like as part of our ClinGen working group. And so it wouldn't, in general, that's like we won't do it. We're not planning right now to do it broadly, as Ryan was saying, but I think there might be some very specific targeted ones that we would do it for.
That makes a lot of sense. And so, do you think at this point, if a VSEP, for example, is really running into trouble classifying one of these types of variants, would it be okay if they reach out to you, like for
Yeah,
consultation?
please.
To
We'd love that.
Okay,
Yeah. That's
yeah.
that's
We
we're
don't
we're
charge
actively
much, really.
yeah. As I mentioned, we're actively working through variants that we know are sort of at this bubble
Hmm.
to sort of like test some of our ideas on the framework and how to sort of modify the upcoming one. And so more examples, the better, for sure.
Perfect. Yeah, I just wanted to throw that out there since this group is actively curating and has many coordinators on it too, that
Yeah.
they should definitely reach out.
And, and,
Okay, we
and
have an.
let me add that a lot of our current work surrounds these low penetrance variants. So if
Mhm.
there's variants that you have that you're struggling with or whatever, now would be the time to reach out to us. And we're even having people present different variants in different disease areas. So they could present that and just have a discussion, within the context of our monthly call, or we can connect just separately from the call.
Yeah, that would be great. Maybe with the meeting recording, we can have Kaizong stick at least Marcie's email or a general email into the summary email so that people can reach out if they have that.
That'd be great.
Yep. We always go on the website, I think, too, to
Yes, yes.
find about
Okay,
these publications that are
so
published. Yeah.
next one is about in silico tools. Could retraining in silico tools on low penetrance variants lead to increased conflicts of benign and pathogenic classification since wild type and variant function could overlap in many ways, but only in one pathway lead to disease for the low penetrance variant?
Well, I guess is the nature of the question that now that same tool is being used for both high penetrance and low penetrance variants. I don't know. The idea is to have a specific training and a specific cutoff for low penetrance variants in that gene. Imagining that those low penetrance variants are acting through the similar mechanism. So hopefully that would lead to more correct classification or, more correct provision of evidence.
And it's a little catch-22 because
Mm,
people aren't trained on low penetrance, but we don't have a lot of low penetrant variants that are gold standard to give the tools to
to
train
train
on.
them on.
So it's
Yeah. Mhm.
it's a little confusing. So that's why we want to just understand how they're being represented by these tools and if the tools are like what they're saying about them. So that's just our first step. And it's for computational and functional ones too. It's same with functional data.
Yeah, that's
For
great.
broad for functional data, I mean like MAVE, like broad
Hmm.
assays, looking
Hmm.
at lots of variants.
Great. We have like one more minute. So does anyone have any?
There's a question also about hypomorphic
Oh,
variants, and
yes. Hmm.
yeah, so as Ryan was saying, we're hypomorph is like a very specific concrete term, and no, not everything is going to be low penetrant or risk allele. Some of those definitely are more highly penetrant, depending upon the disorder. And we just didn't. There's a lot of confusion in the field. I think in a lot of the cancer predisposition field, they've been using hypomorph for a long time, and we want to. Part of the reason we didn't want to use that is it's not. All those can be quantitative in terms of like the functional assay. It's not necessarily representative of the biological. Um, Penetrance. And so we are acknowledging that it's there, but it's not sort of
Hmm.
something that
Yeah.
we're approaching. It's yeah.
Yeah, I think the term originally came from like fruit fly genetics. So it was a partial phenotype. And then lately the way we've heard it used or people have been using it has to do with reduced functional assays. So that's how we talked about it here because that's what we've kind of been hearing. But again, yeah, not a term that we're putting out there. But since we're closing up on the whole presentation here, let's just say one last time, we really want everyone to, you know, use these terms and classifications as much as possible to promote harmonization. And as we said, we're available anytime if you want to reach out.
Great. Thank you so much. Well, this is a really wonderful presentation and discussion. So thank you, Ryan, Matt, and Marcie for coming to our call. And we will end this one. Everyone, thank you.
All right. Thanks, everybody.
Right.
Bye.